So let's call this a little title, From Genes to Proteins. How does that process work? And it involves two important sort of sections or two important divisions. But before we get into all that, let's just get our heads around some of the basic concepts. Okay, so we know that we have DNA, right? And the strand of DNA has these little letters that we use to represent the order of the nitrogenous bases. Oh, sorry, 1 second. Okay, so the little bases, the G, the C's, and the T's, and this code that goes along here, right? And remember the complementary nature of these on the corresponding strand. You'll always have a G and a C together and an A and a T together. So the corresponding strand would look like this, okay? A T and a G and a A here, and so on. That's how it works. And we know already from grade 11 that these letters somehow spell out the instructions for the genes. Now, these letters are nitrogenous bases. It's the order of them that matters. And what we've discovered is that there's a direct correlation. There's a very particular sequence of letters. A sequence of these bases has been discovered to code for a specific polypeptide. Polypeptide. A polypeptide, you'll recall, is really just the primary structure of a protein. It's many little amino acids joined together. And so if you make a polypeptide, it can then all of a sudden start folding on itself and create a secondary, tertiary, and quaternary protein. So when we say making a polypeptide, we're basically making a specific protein. Once we figured this out, we started to look at, okay, well, how do the instructions spell out? And it turns out that the letters or the bases form 3-letter combinations, or we could call them the words of the genetic code. 3-letter combinations or words that indicate or correlate with, maybe is another way to put this, that means to sort of be associated with, right? Or correlate with a particular amino acid. So 3 letters of the DNA code, 3 nitrogenous bases in a row, stand for a particular amino acid. And we're going to see how that works when we get into the process. The other thing that's important is to remember that what we call the frame sequence, the order of reading them is important. So I want to just show you what that means by using an English example. If I wrote this sentence, the 3-letter word, big dog ate the cat. Right? The big dog ate the cat. All 3-letter words, because that's in genetics, every word is 3 letters long. The frame sequence indicates the groupings of the 3 letters. Because if you rewrote this and you grouped it like this, just by moving everything sort of over one, and then you'd have this AT and have a missing one at the end. It does not spell or say the same thing. It becomes gibberish and it's unreadable. So the frame sequence refers to getting the right 3 letters in order. So by comparison, if you have a piece of DNA and it says A, C, C, T, A, T, T, G, let's put a C on the end. So I have 3-letter. There are 3-letter words that form. There are 3-letter words that form. Okay, you want to go back for a second? Or words, genetic words that indicate or correlate with a particular amino acid. It's important that the DNA be read like this. Those three, and those three. You can't just arbitrarily start at the C and go CTA, TTG, because those words are gibberish, just like the words up here. So the frame sequence means it has to be read in a particular way so that we get the right words. Okay? And then we can figure out what the genetic sequence is. Now, what we've been able to do is we have been able to take a piece of DNA, and it took a long time, but we were able to figure out what each letter stands for. And it turns out that each letter forms what's called a codon, right? So these genetic sequences of 3 bases are sometimes referred to as codons. Codons, because they code for something. They code for a very specific amino acid. So that's another word for these 3-letter words, codons. Okay. Now what I'm going to do is I'm going to show you what we've discovered by using the internet. We'll go to the internet, and I'm just going to Google here mRNA codons. And I'm going to show you a chart, which is the easiest way to do it. Okay, this chart shows you there's 4 and 4, so there's 16. But you'll notice some of them have doubled up. So essentially, this chart is showing all of the, remember the amino acids? There was serine and phenylalanine. Well, they're shown here. I think I still have my, they're shown here like right there. Phenylalanine, leucine, serine, tyrosine, right? Arginine. They're all shown there. Beside them are these codes, these 3-letter codes. Now, these are codes on an RNA molecule, which I'm going to explain in a minute, which is why they have U's in them. And what happens essentially is that the DNA is going to open up and an RNA is going to be made along one of its strands. And that RNA is basically the messenger that's going to bring the code to the ribosomes for processing into a protein. So now I'm back on. So what I said was that each of these 3-letter codes, I'll do it again here for those who couldn't hear me. Here is a 3-letter code. All these 3-letter codes, GCU, for instance, would stand for alanine. And GUU would stand for valine, and so on. And each amino acid has a code. What you'll notice, though, some of them have more than one. There's redundancy built into the system. Is that what you were going to ask me? Yeah. Like, look at valine. There are 4 different sequences that can code for valine. We don't really know why this is. Well, we suspect it's because, remember, the evolution is a messy process, and it's never just, like, perfectly lined up. These things evolved over time, and what this might be is an indication of the advantage of having these 4. So if you look at them, they're all very similar. So GUU, GUC, GUA, and GUG, right? I'm going to write those down, and then I'll show you what I mean, why that would be advantageous. So let's go back to our, right? If you have GUU on a piece of DNA, but you get a mutation where the U actually on a piece of RNA, where the U gets torn off or something happens, right? Um, and another one gets put in, it gets replaced by, say, an A, well, it doesn't affect anything because it still codes for the same amino acid. So this redundancy allows for some of the mistakes that might happen in the DNA sequence due to the actions of other molecules, free radicals attacking the molecule, ultraviolet light that can cause electrons to move and jump and wreck the molecule. Sometimes the mistake will have no effect whatsoever, so it's kind of like a redundancy. What other things would you think would be coded for? Like starches or yeah, no. The DNA sequence codes for amino acids only. And in our next video or the next couple of videos, we will see exactly how that works. Right now, I'm just letting you know about the 3 letters. But it involves the actions of 3 different kinds of RNA, right? And so just to wrap up, the process we're going to involve is mRNA, which is called messenger RNA. Messenger RNA. And it is the molecule that copies the information from DNA. So maybe you could write that, okay, copies from the DNA. Then there's something called transfer RNA, or little t RNA. And we use a small letter and then 3 capital letters. Transfer RNA. Transfer RNA, its job is it has on it a codon recognizer sequence, and it carries amino acids around with it. And so it's like the taxi that brings the amino acids and places them in their right spots to be made into the polypeptide. Carries the amino acids. And then, lastly, we have what's called ribosomal RNA, or rRNA. Ribosomal RNA. And this is RNA that is bound with proteins in the ribosome. It makes up the ribosome structure. And serves as a binding site for the messenger RNA that's bringing the message. So the messenger RNA collects the message from DNA, breaks free, has to have a few modifications made to it so it can get through the nuclear membrane. And then it travels out of the nucleus to the ribosome where it attaches and waits for the transfer RNA to bring in the amino acids. But we'll get into all that in detail in the next video.